So it's really, as always, a great pleasure for me as president of the Global Virus Network to welcome all of you to the GVN webinar series at the forefront of virology and on COVID-19. Great pleasure to welcome the lecture of Richard Scheuermann. As indicated on the slide, Richard is a professor and director at the Craig Venter Institute at the Jolla Campus. He's also adjunct professor at the University of California. He's the director of one of the GVN Centers of Excellence and a member of the Global Virus Network SARS-CoV-2 Task Force member. So first, I just want, before introducing Richard, to remind everybody what is the GVN about. I know that most of you are aware of it, but some are not. So Global Virus Network is a network of now 57 centers of excellence worldwide and 11 affiliates. It's really a science-driven and independent network which really aims to merge the best scientists worldwide to get their expertise and to get their scientific input on all actually human viruses. So it's about a scientific project. We have annual meetings also which gather everybody from the GVN. We have a task force. Obviously now we have a task force on COVID-19. We have a lot of educational and training activities, a lot of advocacy statements. Also, GVN has many partnerships with industry. We have created with Lynn Manley, the vice president, what we call the GVN corporate centers, which allow companies and biotech to really be part of the strategy of the Global Virus Network. So, and next week, actually, we will have the GVN annual meeting, which will be an online meeting, which will be a closed meeting for GVN members, but we will have on Thursday a press conference. And the idea of the workshop will really to provide insight and update as to what has been wrong in the way we have handled the crisis, but what has been working and which lessons we can take for the future of handling this epidemic and the future pandemics. Okay, so this is GVN, and this webinar series is part of our will to communicate the knowledge of our best experts. And in this context, again, it's my great pleasure to welcome Richard Scheuermann. Richard has a very interesting career track. He has been trained in the Massachusetts Institute of Technology, MIT. Then he had his PhD at the University of California, Berkeley. Then he spent some time in the University of California in Berkeley, went for 5 years at the Basel Institute for Immunology, very famous institute, where a number of very high-profile immunologists had been trained for many years, and then moved for 10 years from 1992 to 2012 to the University of Texas Southwestern Medical Center, and he became an assistant professor, then associate professor, then professor with tenure, and then moved to, again, back to California. He went to the, and he became actually part of the Craig Venter Institute, and he's now professor and director at the Craig Venter Institute. He's a great and highly recognized expert on bioinformatics and a number of items. And I would also close by telling that Richard has been a great support to the Global Virus Network, always a very active member and a friend. So it's a great pleasure to welcome Richard and to listen to his lecture. Thank you, Richard.
Thank you, Christian. Thanks for that kind introduction. I appreciate it. And yeah, the Global Virus Network is definitely a priority activity for me, and I really enjoy being an active member of this consortium. So I view myself as a computational biologist, And as such, computational biologists are really enablers. Our goal is to really enable people, investigators, to use computational methods to help kind of address their own research questions. And so what I'm going to talk to you about today is a database resource that we developed to support virology research called the Virus Pathogen Resource, and how we've been using that to help support COVID-19 research. So let me share my screen. Okay. All right, I assume everybody is seeing my screen. Okay, so much of our research in virology is supported in the United States by the National Institute of Allergy and Infectious Disease. And you are probably familiar with Tony Fauci, who is the director of that institute. One of their programs is a genomics program that is designed to produce kind of high throughput data through genomic sequencing centers, through proteomics centers, systems biology centers, structural biology centers. And so we have these programs that are generating kind of massive amounts of data related to infectious diseases. And so several years ago, NIAID decided that they wanted to have a database resource that aggregates and disseminates all of this data that's being produced by these genomics programs. And so they initiated what they called the Bioinformatics Resource Centers program. And the goal of that program is really to disseminate all of these data and make them easily accessible and usable by the research community. So they funded 5 different bioinformatics resource centers that are focused on different collections of human pathogens. And there are actually 2 centers focused on viral pathogens, the Virus Pathogen Resource and the Influenza Research Database. And my group leads the development of both of those resources. So these are kind of screenshots of the home pages of the 2 resources. They actually look quite similar, and in fact, they're built on the same basic computational infrastructure on the back end, and the front end is just more tailored towards the specific uses and needs of the different research communities. But the goal is really to provide this comprehensive integrated database about human virus research and surveillance data. The resources are free and open access with no restrictions. So you can download all of the data, you can use all the tools, however you prefer. We really focused on doing things like data curation and data standardization so that you get consistent results when you're using the data in these resources. And so not only do we kind of aggregate data from a variety of different kinds of data sources, but we also try to integrate those data in useful ways. And then we also generate novel data that are available through these resources using computational pipelines and methods that our group has developed. In addition to the data, then we also try to provide access to analytical tools and visualization tools to allow people to explore the data in useful ways. And currently we have our infrastructure built upon high performance computing resources at Argonne National Labs, which is one of the national labs in the United States that have, large compute facilities that we're able to access. Users of these resources can set up their own private workspaces in our workbench function. And so, that allows you to utilize all of these tools and save your results, but also to upload your own kind of private data and integrate those data with all the public data that we make available through these resources. There are, this is just kind of a summary of the data that's available in the two resources, and I'm not going to go through this in detail, other than to say that we really actually have kind of three sources of data in IRD and ViPR. One is that we aggregate data from public data archives like GenBank, UniProt, the Protein Data Bank, Immune Epitope Database. And so we're bringing data in from those data archives. And as we're doing that, we're curating the data and standardizing the data, especially the metadata, so that you really get kind of consistent representation of things like host species, country of isolation, dates, and things like that, so that all of that is kind of standardized and curated. We also get data via direct submission from specific programs supported by NIAID. So you may be familiar with the Centers of Excellence for Influenza Research and Surveillance, the CIRS program. And so all of the surveillance data that is generated by that program comes into IRD through direct submission from CIRS. And then the third source of data are these novel data types that we derive, through our own computational pipelines. This is an example of the search interface that we have developed in order to allow investigators to access the data in the database. And each one of these search pages is customized to the type of data that are being interrogated. This is an example of the search interface looking at influenza proteins, and you can see that you can search based on different host species, different geographic locations, and in this case, if you're searching for influenza proteins, you can search for the classical proteins like HA and NA, or variant proteins. These are novel proteins that have been identified in influenza, like PB1F2 and PAX and things like that. And each of the data types has kind of this custom search interface built around it. In addition to the data, then we also have a whole suite of different analysis and visualization tools, including ability to blast against different subsets of data in the database, and a whole variety of different comparative genomics analysis tools. And our goal is to really make it very seamless to identify data sets using these search interfaces, and then take those into these analysis and visualization tools in a very kind of seamless workflow. And I'll show you a couple of examples of that a little bit later. And then finally, I mentioned that you can set up your own private workspace, and this is a screenshot of my workbench. And this allows you to save your data sets that you might pull out of the database, save your analysis results, and also upload your private data and integrate that with the data that's available in the database, and then be able to use all of these analysis and visualization tools on your own data. So we actually have, these resources are used by the international virology community. This is just a plot of some of our usage statistics starting in January of 2019. We get about 4,000 to 5,000 users per month of each of the resources. And this is a very typical phenomenon that you're seeing here in the setting of an infectious disease outbreak. So, when all of a sudden people started to learn about this outbreak of novel coronavirus in Wuhan, what we see is this big uptick in usage of our resource, people looking for data about the virus and looking for use of these analysis tools to be able to explore the genomic characteristics of the virus. And so, what we saw in the case of the coronavirus outbreak is, a fourfold increase in usage within a matter of months. This is in kind of gray and yellow here. This is usage of the influenza research database. And I think this is kind of a curious phenomenon that, people for some reason also were using the influenza research database, at increased frequency even though there's really no data about coronavirus in that resource. It's kind of an interesting phenomenon. And then typically what we see is that the usage will drop again. But one of the things that we typically notice is that when it does drop, it reaches a new plateau of kind of consistent usage. And in this case, we get a 50% increase in just kind of consistent usage of ViPR once people learn about the resource in the context of these types of outbreaks. So this is the ViPR homepage, and the URL is listed here. It's www.viprbrc.org. We support data about 14 different families of viruses, and the families are selected because they include human pathogenic viruses. So we have data about coronaviridae, flaviviridae, picornaviridae, phyllo, pox, herpes, rhabdo, all of the major classes of viruses that infect humans. And then in addition to the human viruses, we also bring in viruses from other host species that are related to the human pathogenic viruses to help support comparative genomics and understand, animal reservoirs and how animal viruses jump into humans and things like that. And so, these are the families of viruses that we cover. In addition, whenever there is a high priority virus that might come out associated with an infectious disease outbreak, we develop these featured virus portals that allow you to easily access the data about these high priority viruses. And so, when the coronavirus outbreak occurred, we quickly put together a portal into the data about the SARS-CoV-2 virus. So since the COVID-2 outbreak, we've added a number of enhancements to ViPR, and I just wanted to highlight those because they're all tailored to support research around coronavirus. So first of all, I mentioned that we developed this portal that makes it easy to access all the COVID-2 data. This is a screenshot of the homepage of the SARS-CoV-2 portal. We have some basic information about the virus, and then this allows you to very quickly access all of the data related to the virus. As of this month, we have about 13,000 whole genome sequences for coronaviruses, and as well as all the protein sequences that are encoded by those genomes. We have link outs to a variety of different other resources, including reagents at the BEI resource, epidemiological data at NextStrain, and link outs to other kinds of useful resources for coronavirus research. And we basically put that portal together and released it in February of 2020, so pretty rapidly after the outbreak started. The second enhancement that we added, when we were looking at the genomic sequence data that were being deposited in GenBank and in GISAID, what we found was that the protein annotations were very inconsistent. And so what we decided to do was to develop an annotation tool that's based on a software package that we had developed at JCVI many years ago called Vigor. So that as we're bringing all the sequence data into the ViPR resource, we then process it through this protein annotation tool so that all of the sequence records have a consistent and comprehensive protein annotations associated with them. And so in the case of COVID-2, that includes all of the open reading frames, all of the ORFs that encode proteins in the genome, as well as all the mature peptides that are generated through the proteolytic cleavage of these large polyproteins that are encoded in the first basically 2/3 of the genome. And so this is a screenshot of all of the protein annotations available for one a whole genome sequence that includes all of the NSPs, the mature peptides from the polyproteins, as well as all of the ORF sequences. And so if you go to ViPR now, you can get a consistent collection of protein sequences for all of the genome sequences that are available in ViPR. In addition, if you've just generated your own coronavirus sequence, you can upload that into our Vigor4 implementation and generate all of these protein annotations for your sequences and download those in a variety of different formats. And if I have time, I'm going to try to demonstrate the use of that tool in a little bit. The third enhancement that we added was a tool that can take next generation sequence data using two of the major sequencing platforms, Illumina and Oxford Nanopore, and basically take the FASTQ files that come out of those sequencing platforms and do reference-based assembly into genome contigs, taking the reads and assembling them into contigs, and then annotating those using the Vigor4 tool so that you get the comprehensive protein and peptide annotations, and then also doing a variant analysis to look to see the extent of sequence variation in comparison to a reference sequence. And so now this is available as a service at Argonne National Labs. And so you can either upload your FASTQ files, or you can extract, if you've already uploaded those files to the sequence read archive at NCBI, you can extract those sequences from SRA and then have those sequences assembled and annotated using this pipeline. And then finally, in order to really understand the whole family of Coronaviridae and the differences between Coronaviridae that circulate in animal reservoirs from those that circulate in humans, we've started to add in a detailed evaluation of the protein orthologs across the entire family of viruses. And the way that we approach this is that we examine the domain architectures of the individual viral proteins to get a sense for the large scale structures of these different proteins. This is actually going to, I think, be quite interesting to understand the differences between viruses that infect different hosts. This is a schematic of the domain architecture of the ORF1ab polyprotein in the different genuses of Coronaviridae. And what you can see is that the carboxy terminus of the polyprotein is pretty well conserved across the entire family. So starting with NSP3 and moving down, you see the domain architecture of that polyprotein is very similar, regardless of which genus you look at. But if you look at the amino terminus of the polyprotein, you see there's a lot of very interesting differences between the different genuses of Coronaviridae. And I think this information, we're in the process of releasing all of this information about the domain architectures of the Coronaviridae proteins through the ViPR resource for people to be able to explore correlations between these different domain architectures and other virus properties. So that's just a quick overview of the ViPR resource and what we've been doing recently for support of coronavirus research. What I wanted to do now is actually do a live demonstration of the resource just to give you a sense for how you can use these interfaces and tools to explore the data that's available. So let me pull up my browser. So here is the ViPR homepage. I think about the resource as kind of having these three major components. You have the data that's accessed through these search interfaces. You have the analysis tools, and then you have the workbench feature that allows you to share and save and share your own data. I already mentioned that we support data about the 14 families of viruses and we have these featured virus portals for easy access. In addition, we try to keep up with the latest news in virology and interesting reports and articles that are published. We will highlight in this virus news area. Also, when there are meetings coming up, we'll post information about those meetings here, etc. And then we're actually quite active in social media, so you can sign up to receive notifications and information through Facebook and through Twitter. We also post tutorials in YouTube. We have a YouTube channel, and you can also sign up to be on our mailing list. And so every time we have a new release with new features, we send out a newsletter that kind of highlights anything that's new that we have implemented in the resource. So this is kind of what the home page looks like. So I mentioned that, you know, people access the data through, let's go ahead and go to the Coronaviridae homepage. And so this is just kind of a summary of the different types of data that we have in the resource, and then links to the search pages to access those data. So we have genome sequence, protein sequence, and strain information. We have information about immune epitopes that have been defined in the different viral pathogens, information about the different ortholog groups that I was just describing, information about antiviral drugs, host factor data that looks at host response to virus infection, like transcriptomic and proteomic data, three-dimensional protein structures, and information about protein domains and motifs, etc. And so then if you click on one of these links, now it takes you to the search page, and this is an example of one of the search pages. This is to launch kind of a genomic search. And what you can see here is these are the different criteria that you can use to search for different genome sequences or protein sequences. Actually, what I wanted to do is, let me go back. I am going to go to this CoV-2 portal page and again, launch the genomic search. Okay, so again, this is the search page. What you see here is that as you're kind of formulating your search criteria, we quickly compute the number of records that will be returned based on those criteria, and we make that available in real time up here. This is actually a kind of a useful feature so that you get a sense for the number of records that you're going to return based on the search criteria that you select. So in our case, if we say, okay, we're going to focus our search on isolates from 2020 and we're only interested in human, what you'll see is that now these numbers start to change based on these search criteria. And let's say that I'm only interested in complete genome sequences. I don't want to pull out partial sequences. So there's an option of searching for only complete genome sequences down at the bottom. There's also a whole series of different advanced options down here that allow you to search based on month ranges, using public identifiers, other kinds of search options for more advanced kind of search queries. But if we just kind of use these basic criteria here and then press the search button, now it's going to go into the database and retrieve records based on those criteria. And then initially what we do is we produce kind of a summary table that provides some basic information about the sequence records that are returned based on those criteria. And so this is kind of a summary table that has some information about each of these sequence records You can change the display of this table by coming up here to this display settings option. And for some reason, it's a little bit sluggish, but so you can choose which display fields you want to include in this table. You can sort the records in the table based on different criteria. You can choose to have more items displayed on the table. And so it allows users to kind of customize the look of these summary tables that are generated from these search criteria. So then if you want to learn more about each of these individual strains, you can come in and click on this view link. And now this takes you to a strain details page that has more detailed information about the strain. It has information about the genome sequence associated with that strain, isolation source, other kinds of information. This then links to the GenBank record from which the original data was derived. If you click on this link, you can view the nucleotide sequence for that genome. And here's an example of where we have this kind of close integration between the data and the analysis tools. So let's say that you're interested in designing PCR primers for this genome sequence. If you click on this link here for PCR primer design, this takes you to a primer design tool that's based on the Primer 3 software. The genome sequence data is loaded for you. And then down here, you can then basically configure the Primer 3 tool to include or exclude particular regions of the genome. You can design the primers that will generate products in different size ranges, have PCR primers that have different sizes and different melting and GC content characteristics. So you can really configure this software package to pull out PCR primers, that are very much tailored towards your specific needs. Another nice feature of our database is that we have up here what's called breadcrumbs. So we're basically keeping track of, the steps that you're using as you're migrating through the resource. And so if you then want to go back to the strain record or the search results, you can just click on the breadcrumbs and it takes you right back to, where you were in that kind of exploratory workflow. So as we scroll down further, we have this nice kind of schematic of the genome. And the protein coding regions. And then down here, this is an example of the consistent protein annotations that I mentioned earlier. So for this particular sequence record, the GenBank record has information about only 5 of the open reading frames encoded by this genome. So ORF1AB, the surface glycoprotein, the envelope protein matrix, and nucleocapsid protein. So these are the annotations available in the GenBank record. But then as we brought that sequence into ViPR, we ran it through our Vigor4 annotation tool and produced all of the mature peptide and a comprehensive annotation of all the open reading frames. And so all this data is then available in the resource. So, that's an example of, what the sequence records look like in ViPR. We have this feature here where we save the search criteria that you were using. And so now if you click on kind of the search criteria here, and maybe if I go over here, you can kind of go back and adjust the search criteria that you've used and make some adjustments to kind of change things up a little bit. Sorry. All right, so for example, let's say that instead of looking for all human sequences, we want to restrict our analysis to sequences that are isolated from particular countries. Let's say we're interested in exploring a question about, are there differences in the sequences of coronavirus isolates that are isolated from different countries? And so we might be interested in, say, looking at what kind of sequences are found in different countries within Asia. And so if we come over here and select Asia as our geographic grouping, and then come here, and let's say we want to compare sequences from China, and then also say Japan, and also Thailand. And now instead of doing a comparison of genome sequences, I want to look at a particular protein. So I click the protein button, I come down here, and then I can basically search based on protein, the gene symbol or the protein product name. And so let's say that I'm interested in the RNA-dependent RNA polymerase to see if there are any significant sequence differences in the RNA-dependent RNA polymerase between these different countries. And so I can go ahead and kind of launch that search. Oh. Okay, that was weird. Let's try that again. Okay, so now if we change this to protein, RNA-dependent RNA polymerase, now you can see that there are 149 records that match those search criteria. We click on the search button. We get this summary table now, and you can see that these are all now gene products for RNA-dependent RNA polymerase. And so now what I can do is select all of these records. My internet seems to be pretty sluggish today. Ooh. Sorry about that. It is doing some weird things. Well, I'm just gonna skip over that, and I will kind of come back to that again later. So one of the other things that I want to kind of quickly demonstrate is one of these tools that I mentioned earlier, the Vigor4 Genome Annotator. So what you can do here is, if you have your own CoV-2 sequence, genomic sequence, and you want to generate this comprehensive set of protein annotations, you can come over here and you can upload a file or paste in the sequence in a FASTA format, and then run Vigor4 to produce the annotations. And so in this case, I will choose a sequence file, a FASTA file. In this case, this was a sequence that I downloaded from the GISAID resource, which does not have any of the protein annotations in it. And so I can upload that record into the Vigor4 tool and then click run. And now it will generate these protein annotations. Now, whenever you launch an analysis job like this, you'll see this screen. And if the job is going to take, an extended period of time, you can choose to not wait here for it to finish, but you can save the analysis in your workbench so that it automatically gets saved there, and then you can go and retrieve it at your leisure in the future. And you can also request notification from when this analysis task has been completed so that you receive a notification by email. So I actually ran this. This task takes about a minute or two, but I ran this previously and I chose to save the analysis result in my workbench. And so now if I go to my workbench here, I can come down here, and here is the results from that Vigor4 annotation of the GISAID record. And so I can click to view that. And now here you see that this is the annotations that were produced for that particular sequence. So that's an example of one of these kind of analysis tools and how you can use that and save the results to your workbench. So that's just kind of a quick overview of the ViPR resource and the kinds of things that we've done specifically to support coronaviridae research. I thought what I would do is just kind of end my presentation today by talking a little bit about the science of coronavirus. And so let me go back to my PowerPoint. So one of the questions that we're interested in monitoring is, how is SARS-CoV-2 evolving as it is migrating through the human population? And this is an important problem when we're dealing with zoonotic viruses that have an animal reservoir that then spill over into humans and then adapt to their new host species. And so typically what you will see is that there will be sequence substitutions that are associated with the initial viruses that are able to jump into humans. And then as they're transmitting within the human population, they will acquire additional sequence variations that allow them to become more fit in their new host population. That might increase their transmissibility and other kinds of characteristics that allow them to be more efficient at replicating and transmitting within their new host population. And so, we can use the genomic sequence data and the analysis tools in resources like ViPR to explore this question. Is the coronavirus evolving? And is there evidence that specific variants are being positively selected due to enhanced fitness of the virus? So in this case, we've taken all the sequence records and we have analyzed them to understand the extent of sequence variation that we see at different positions along the genome. And in this case, we're using an entropy score to get a sense for the extent of variation in the genome sequence record. Entropy is a pretty common way of quantifying sequence variation. This is the equation here. And what you see here is that there are these positions where we're seeing high entropy, so high sequence variation that suggests that novel variants are being produced and their prevalence has been increasing over time, or that they're existing in significant prevalence within the population of viruses. And so when we looked at these particular high positions with high entropy scores, we saw three different kinds of patterns in terms of the change in prevalence over time. So the first pattern that you see when you look at that, and these are two particular substitutions that result in non-synonymous changes in the NSP6 and the ORF3A proteins. These kinds of substitutions, we think, show neutral evolution. They're neither selected for or against. And the reason that we think that's the case is that if you look at the change in prevalence of these substitutions over time, so these are now the number of records or the proportion of records that have two different sequence residues at that position, you see that this new variant appears, but then its prevalence just kind of sticks around and it doesn't really change very much. It's pretty flat. And this is the kind of pattern that you see when there's really no fitness advantage or disadvantage to the substitution. It's relatively neutral. And so for a certain period of time, you'll see that both of these variants will co-circulate in the host population. In contrast, this is an example of substitutions that we think are undergoing positive selection and that result in increased fitness of the virus as it's circulating in humans. And this is kind of the pattern that you see for positive selection, is that the prevalence, so you have the emergence of a new variant at a certain point in time, and then the prevalence of that variant increases over time so that it then at some point becomes the predominant species of virus that you see circulating. And so there are two examples of this kind of pattern. One of them is within the S-glycoprotein, this D614G substitution that you probably have heard about. And another one is in the RNA-dependent RNA polymerase, the P323L substitution. So in the case of this S-glycoprotein substitution, this is a schematic of the structure of the glycoprotein. Initially, we thought, well, maybe this substitution affected its ability to bind to the cellular receptor for coronavirus, but it turns out that the receptor binding domain for the ACE2 receptor is located here. And the substitution that we see that has kind of become predominant is actually not located within the receptor binding domain, but it's located within this subdomain region here. And actually, if you map these regions onto the three-dimensional protein structure for the glycoprotein, in green is the receptor binding domain here, and in red is this new substitution that we see that has gained in prevalence. So it doesn't seem to be directly affecting host receptor interaction, but there are two papers have been published recently that have explored the functional consequence of this particular substitution. And in one of the papers, they found that patients that had this new amino acid, the glycine at that position, actually had higher viral loads in patients in comparison to patients that had the original aspartate amino acid at that position. And then in this paper, they found that the glycine substitution showed more efficient cleavage by the elastase 2 protease that kind of cleaves the glycoprotein between the S1 and the S2 subunit. And that seems to result in increased infectivity in cell cultures. And so even though the substitution isn't located within the receptor binding domain, it does seem to allow the virus to infect cells and maybe produce higher viral titers in infected patients. So this is going to be something interesting to continue to evaluate, but this looks like it has been positively selected to make the virus more fit for circulating in humans. In the case of the RNA-dependent RNA polymerase substitution, this part over here is the domain that binds to the RNA during replication. And this is the structure of the polymerase as it's replicating. Here is the double-stranded RNA that's generated as part of that replication process. But the substitution that we're seeing that seems to have become predominant is actually at this interface region here. It's actually not in the part of the protein that's actively engaged in replication, but it's in this interface region, and it looks like it may potentially impact the interaction between the RNA-dependent RNA polymerase and this NSP8 accessory protein that's also involved in the replication complex. And so I think it'll be interesting to explore whether this substitution impacts replication efficiency and perhaps the efficiency of interacting with this NSP8 accessory protein. But that's a hypothesis that I think is worth exploring. And then this is the final trend that we've seen, the final pattern that we've seen for this particular substitution here. And this is actually a series of 3 tandem substitutions in the nucleoprotein. And it affects actually 2 adjacent codons and results in 2 non-synonymous substitutions in adjacent codons. This particular substitution is not located in the RNA binding region of the nucleoprotein, but immediately adjacent to the RNA binding protein. And the fact that one of the substitutions adds a basic amino acid means that it perhaps could extend the RNA binding region of the protein by adding this additional basic amino acid and maybe extend this interaction interface between the nucleoprotein and the RNA molecule, the genomic RNA. Again, that's a hypothesis that's worth exploring. And it'll be interesting to see if the prevalence of these tandem substitutions increases over time as the virus continues to circulate in the human population. So, this is, summary of the bioinformatics resource that our group has developed to support viral research, including coronaviruses, and some applications about how one can use the data and the tools to explore questions about virus evolution for SARS-CoV-2. And I'll just close by mentioning the fact that as part of our mandate of building these resources, we engage in a variety of different outreach activities where we train people on how to use these kinds of resources and these kinds of comparative genomics analysis tools. Over the years, we've hosted close to 100 different workshops all over the world that range in a length from 1 day to 4 days, and are tailored towards the needs of the groups that we work with to organize these different workshops. And we've done several of these together with the Global Virus Network and at particular GVN Center locations. So in the future, if groups are interested, we would be happy to organize these kinds of outreach activities, and we just would need to kind of collaborate about that. But obviously right now we're not doing any traveling because of COVID. And so currently what we're doing is we're doing workshops through virtual webinars. And this is a schedule of webinars that we've been doing recently. There are several that are coming up in October and November and December. And if you go to the ViPR homepage, you can register for attending workshops that are on different topics. And the workshops that we're doing this year are obviously targeted on coronavirus and SARS-CoV-2. This is the team that's been involved in developing ViPR and IRD, my group here at JCVI, really a talented group of bioinformatics analysts and engineers. We're working with investigators at the University of Colorado and Argonne National Labs that maintain the compute resources that we utilize, and then collaborators at the University of Virginia and the Fellowship of Interpretation of Genomes are all involved in the development of the ViPR and IRD resource. So with that, I'll stop and happy to take any questions.
Thank you so much, Richard. Thank you for a great lecture, which really demonstrates the benefit of such facilities and expertise and very interesting results on COVID-19. So I remind the audience that you can share your questions, and Richard will have the capacity to read them. So you can start to. So I don't see presently questions so far. So maybe just to start with the last part of your presentation regarding the 614 mutation, I mean, in the spike, I do understand the impact on contagiosity and infectiosity, but in the paper that you have been referring to, the recent one, they claim decreased neutralization sensitivity. But as I understand from your point, I mean, there is really no direct effect on the immune impact. I mean, it would be really more by the infectiosity. Is it true? Or?
Yeah, you know, that particular finding is controversial. There are other groups that have, you know, looked at serum reactivity and not seen any differences between spike proteins that carry these different substitutions. So I think, to me, the jury's still out on that aspect of the virus. And so we'll just have to see. You know, keep in mind that, you know, when you get a serum antibody response, it's a polyclonal response. And so you have antibodies that are binding to a variety of different regions of the spike glycoprotein. And so it would be surprising to me that a single substitution would have a big impact on the polyclonal reactivity. Mm-hmm. I could see how it might impact a monoclonal binding, but it's hard to imagine how it could have a major impact on polyclonal reactivity. And certainly that's what's been seen by other groups that have looked at that question.
Yeah. And to follow up on this, I mean, because this, the overall database is very impressive, and is there any evidence for difference of distribution of these three? As I understand, I mean, you have really identified 3 parts of the genome where substitutions can really have an impact on the evolution of the virus. Is there any difference between different geographic areas as to the distribution, or?
Yeah, that's a good question. You know, one of the things that I think anybody who's going to do this kind of analysis has to be very careful about confounding factors. And, you know, the emergence of a variant in a particular geographic region, and then the expansion within a geographic region due to a founder effect. That's another way that you can see changes in prevalence of virus because of a single introduction into a region and then expansion within that region. That can also give rise to these kinds of skewings in prevalence. In this particular case, you know, these substitutions here, the ones here that seem to be indicating positive selection, you see these substitutions in many different geographic regions. So it's not due to kind of this founder effect and expansion within a particular geographic region. These things seem to be gaining prevalence around the world. And so, you know, that is something that one needs to be cognizant of and make sure you control for.
Yeah. Thank you. So I see that there is a question. Can you see it, Richard?
No. Where is it?
At the bottom of your screen. I can read it otherwise, but...
Yeah, can you read it?
Yeah. So this is a question from Mark Kaplan. Are there any clues from genomic data as to why some viruses produce asymptomatic infection?
Yeah, that's a good question. So with this D614G substitution, groups have looked to see if there the prevalence of the 2 variants are different in patients with severe disease versus mild disease. And they don't see any difference. So you see both variants in both mild as well as in severe disease. So as far as I'm aware, we have not identified any genomic substitutions that seem to correlate with disease severity for this virus at this point. And if you think about evolution and the evolutionary pressures that might be selecting for particular variants, severity actually is not a good, selecting for viruses that have differences in severity is not something that is a strong selective pressure for viruses. There's really no advantage for a virus to cause more severe disease. In fact, in some ways, it's a disadvantage for the virus, because if you're killing off your host, you're going to be less likely to transmit and spread within your host population. So we're not seeing evidence of genomic variant associations with severity. And that's something that I think is something that occurs outside of kind of evolutionary selection.
Yeah, that's a very important point. Okay, I believe that we have to close the webinar. And yeah, so, well, thank you so much. Thank you so much. That was very informative. And thank you to all of you for attending these webinars. And as we used to tell now at the end of each meeting, be safe. And we will interact. No, there is another question, sorry. We have, yeah. That's a comment, actually. Jose Esparza, very good presentation. Thank you, Jose. Nice to be in contact with you.
Thank you.
Okay, have a great day.
All right. Thank you. Bye.
Thank you, Richard.